Okay, let's move on to quaternary structures of proteins. For quaternary structure, this is exhibited only by proteins that contain more than one polypeptide chain, or in other words, it's a multi-subunit protein. If your protein only has one chain, then it's irrelevant to talk about quaternary structure, or it doesn't apply to talk about quaternary structure. So, if you're talking about quaternary structures, you talk of dimers, trimers, tetramers, and so on. Now, what's the importance of quaternary structure? These types of proteins usually exhibit allosteric behavior. What is allosteric behavior? This is where changes in one subunit influences the behavior of other subunits. We will illustrate the quaternary structure using hemoglobin as an example. Okay, we just finished talking about myoglobin, which is an oxygen storage molecule, and now hemoglobin, which is an oxygen transport molecule. In terms of structure, it's a tetramer composed of two alpha subunits and two beta subunits. Now, sometimes it is referred to as an alpha beta sub 2, meaning the alpha and a beta subunit are actually more intimately connected with each other rather than with the other alpha beta partner. Each of the subunit in hemoglobin resembles myoglobin, or in other words, an alpha subunit resembles myoglobin. So everything we talked about myoglobin will apply to the alpha subunit, and it will also apply to the beta subunit with some minor differences. The minor differences is that the alpha chains are only made up of 141 amino acid residues, whereas the two beta chains are made up of 146 amino acid residues. Again, in contrast, the myoglobin is made up of 153 amino acid residues. So here's a picture of myoglobin. So it's color coded, no? So if you look at, say, this partnership of an alpha and beta, they may be more intimately connected to each other compared to this alpha and beta. Well, actually, I'm not too sure, but that's the picture, no? This alpha and beta are too far apart from each other. So it's probably this that are close to each other, and then this that are close to each other, and then there's two of them. As mentioned, myoglobin is an oxygen storage molecule, whereas hemoglobin is an oxygen transport molecule. So let's compare the oxygen binding behaviors of these two molecules. So here's a plot of the degree of saturation versus oxygen partial pressure. Okay, so please notice the units that this is now in torrs. No? So this is the partial pressure of oxygen in torrs. So for myoglobin, notice how steeply the curve rises as a response to oxygen pressure. Or in other words, myoglobin picks up oxygen quite readily, even if there's just a few moles or moles. Again, reminder, the pressure of oxygen is proportional to the number of moles of oxygen present, okay, the concentration. So notice that myoglobin picks up oxygen quite readily. Okay, so just a few torr of oxygen and it's already bound to myoglobin. And you get almost saturation, okay, meaning all of the myoglobin may have already picked up oxygen at about 50 torr. Okay, so I leave it up to you. Reminder, 1 atmosphere is 760 torr. So you can imagine how small the pressure of oxygen is, and yet myoglobin will pick it up. For hemoglobin, it's not as straightforward. So even if you increase the partial pressure of oxygen, some of the hemoglobin may not pick up the oxygen yet. And then at some point, no, so it now reaches about 50 torr before you reach about, let's say this is about 75, 80% saturation. Whereas for myoglobin, 70-80% saturation of oxygen occurs at about 5. This is about 1 torr. I know, so this is 50%. So 80% is about 2-3 torr. So the curve, to be mathematical about it, so don't be scared. The myoglobin curve is described as being hyperbolic. And for the hemoglobin, the oxygen saturation curve is described as being sigmoidal. Okay, sigmoidal refers to that, like the S shape. Now, so you're familiar with sigma, the Greek letter sigma, which in the equivalent in the Roman alphabet is the letter S. So this curve, okay, in your imagination, should look something like a letter S, hence sigmoidal. And the other property of this is that for hemoglobin, the binding of oxygen is referred to as being cooperative. So what is now the cause of this cooperativity in the uptake of the binding of oxygen? Okay. So if you look at the picture of the heme group in in any of the subunits of hemoglobin, and also in myoglobin, the iron 2 is originally out of the plane of the protoporphyrin ring. It is only upon the binding of oxygen and also carbon monoxide that the iron 2 is pulled towards the plane of the ring. But, however, remember, the iron is connected to the histidine F8, so it pulls the histidine F8 residue. But the histidine F8 residue is not in isolation. It carries with it the whole uh, F helix in the subunits. Now, again, whether it's myoglobin or any of the subunits in hemoglobin. So we can say that there's now a conformational change in the F helix, and therefore also the whole polypeptide chain. Okay, so this is a picture once again. This is the E helix, and this is the F helix. Originally, the iron is out of the plane. Once oxygen binds on this end, then this helix is pulled closer to the hemoglobin, okay, or they tighten up a little bit. Okay, so here's another pictorial. So again, different orientation. This is the F helix in this illustration. This is the F helix in this illustration. So once oxygen binds on the other end, this is the other end, and this end are the same. Okay, so originally the iron is out of the plane of the porphyrin ring. So here it's color coded. Okay. Our usual case is that deoxygenated blood, right, is blue, and then oxygenated blood is red. We'll more on that later. Okay, so once oxygen binds, the iron gets pulled. So it's now on the plane of the porphyrin ring, and then it pulls the F helix. So originally farther, and then it's now closer. Okay, so here's now a comparison in the myoglobin and in the hemoglobin. In myoglobin, the oxygen binding becomes independent of the other myoglobin molecules. I mean, after all, who cares whether there's a conformational change in the myoglobin as oxygen binds? However, in hemoglobin, as mentioned, there's a conformational change in one of the subunits, and then therefore can also cause a conformation, and can also causes a conformational change in the other subunits. So as if you can infer from the illustration, in oxygenated hemoglobin, the two beta subunits are now closer to each other. Okay. So here's a pictorial representation. These are now the two beta subunits. Okay. So this is deoxygenated, and then once this gets oxygenated, okay, so this is the F helix, the E helix, oxygen binds over here. So now these two get closer to each other, and then the distance between them also becomes smaller. So those are the conformational changes. In myoglobin, as mentioned, doesn't really matter. But in hemoglobin, the subunits are close to each other. Therefore, any change in one subunit affects the other subunits. Okay. In terms of proteins, especially allosteric proteins, it's very usual to talk about the so-called T state and the R state. Now, I'll start with the R state because it's easier to understand. R refers to relaxed. And in the relaxed state of hemoglobin, it is more conducive to accept oxygen. In the T state, okay, T stands for taut, T-A-U-T, which is synonymous to tight, T-I-G-H-T. Hopefully I spelled that correctly. Okay, the distance between the two subunits are farther apart, and then this one favors the removal of oxygen. Other conformational change is that there's a rotation of the alpha B1 dimer. So as mentioned earlier, it's more an alpha subunit and a beta subunit are more closely connected to each other rather than an alpha and a beta, alpha and another alpha and a beta and another beta. But anyway, there's a twist of about 15 degrees Celsius, not Celsius, 50, just 15 degrees. Okay, okay. I don't know if you can see it in the figure, but again, that's just information that we should, might be useful to know. We will not dwell too much on that. We'll probably just dwell on the distance now between the subunits. That focus on the space in between over here, the different subunits. We will get back to that later. Okay. Another phenomenon associated with hemoglobin function as oxygen storage, oxygen transport molecule is the so-called Bohr effect. This is after Christian Bohr, who's Niels Bohr's brother. No? So we're more familiar with Niels Bohr, with the Bohr atomic theory. Christian Bohr is a biologist. Okay? Niels Bohr was a physicist. Okay, so the observation was, again, the concentration of hydrogen ions has an effect on the oxygen binding ability of hemoglobin. Hemoglobin binds to oxygen more easily under basic conditions, meaning higher pH. Okay, so what's the normal conditions of the of organisms that have blood? Okay, the muscle tissues, comparatively speaking, are more acidic, and then the lungs usually have higher pH or less acidic. Okay, so there's now therefore an equilibrium affecting the hemoglobin. Okay, so under basic conditions, okay, the hemoglobin, okay, releases the hydrogen. Okay, so it's not part of hemoglobin. The concentration of hydrogen ions is small, so it's more conducive for hemoglobin to pick up oxygen. So that's in the lungs. Once the hemoglobin now travels to the muscles, okay, the concentration of hydrogen ions becomes high enough that it now protonates or hydrogenates the hemoglobin, and the concentration of carbon dioxide also increases. Now, in the muscles, as we will see later, one of the byproducts of metabolism is the formation of carbon dioxide. So the two go together. Okay. So and picking up the hemoglobin picking up hydrogen ion that is not conducive to holding on to the oxygen. Therefore, the oxygen that hemoglobin picks up in the lungs is transported into the muscle. It is released into the muscles where the environment is more acidic. Okay, and then the protonated hemoglobin that also carries carbon dioxide travels through the circulatory system, goes back to the lungs, and then there the hydrogen ion is released because again the conditions are less acidic, so the protonation of the hemoglobin becomes less favored, and then the hemoglobin picks up another molecule of oxygen, and the cycle just continues. Okay, so the Bohr effect graphically. Okay, so here's now a plot of the oxygen saturation curve of hemoglobin as a function of pH, upon changes of pH, and also, of course, the oxygen saturation. So reminder again, the percent saturation, how much percent of the myoglobin and hemoglobin molecules accept oxygen versus the partial pressure of oxygen. Okay, millimeter mercury. Reminder, millimeter mercury is the same as torr. And reminder, 1 atmosphere is 760 torr. Okay, so again, what happens? Okay. A normal pH of blood is about 7.4. So let's start with that. So this is the normal saturation curve, represented by the green curve. Okay, and then we have the differences in partial pressure of oxygen under venous conditions, meaning the oxygen in the veins, and then the arterial conditions. Okay, so reminder about your circulatory system. The arteries have pressure, is the blood that has just come from the lungs and the heart. So the oxygen levels is quite high. Okay, so notice that again, majority of the hemoglobin would now be about 80-90% saturated with oxygen. And then when you go lower, again, so venous blood is now has come from the muscles. No? So oxygen concentration is not quite low. So it's about 40 millimeter mercury. So the level of oxygen has now dropped. But under more basic conditions, notice that the drop in oxygen is much dramatic. It's more dramatic. No? So pH is 6.8. So the lower the pH, the less the hemoglobin holds on to oxygen. Okay. So once again, that's the secret of hemoglobin, one of the secrets of hemoglobin, such that it's able to function as an oxygen transport molecule. Okay, so the summary of the Bohr effect, again, in the lungs, comparatively speaking, the pH is higher compared to actively metabolizing tissue. Therefore, hemoglobin binds to oxygen, and at the same time, hemoglobin releases the hydrogen ion in the lungs. And actually, also, it carries with it's not written here, but it also carries the carbon dioxide, and we exhale it as carbon dioxide. Uh, and therefore actively metabolizing muscle, it has a lower pH due to the production of hydrogen ions as a side product of metabolism. So hemoglobin uh, binds to hydrogen ion, and then it's not conducive to carrying oxygen. So hemoglobin releases oxygen under more acidic conditions. So that's the summary of the Bohr effect. Okay, so how does that work? Okay, I've been talking about the hydrogen ion and protonation. Okay, so increasing the hydrogen ion concentration protonates some key amino acids. What are these? There's the N terminals of the alpha chains. For the beta chains, the histidine 146, which is involved in a salt bridge to aspartate 94. So this is what it means. No? So histidine 146 means the 146th amino acid residue in the beta chain is histidine. And then in I guess this is also for the beta chain. Yeah, it should be. Okay, disrupts a salt bridge to aspartate, which is the 94th residue in that chain. And then once this gets protonated, it now favors the deoxygenated form of hemoglobin. So another lesson here is that oxyhemoglobin, okay, meaning the oxygenated hemoglobin, comparatively speaking, functions like a strong acid. Okay, but don't misinterpret it. It's not as strong as hydrochloric acid. It's just stronger than what you expect it to be. Okay, so another side effect of this property is that hemoglobin itself, okay, modulates the buffering of the blood pH. carbon dioxide. Okay, carbon dioxide. Reminder: pKa is 6.35. Blood pH is about 7.4. So normally it's protonated. The dominant form is bicarbonate. So the presence of carbon dioxide actually favors the deoxyhemoglobin. And such that I mentioned this earlier already, the bicarbonate is transported into the lungs, and then in the lungs, the carbon dioxide is released. Now, there's another important molecule involved in the function of hemoglobin, and that is 2,3-bisphosphoglycerate. You are given the acronym BPG. This binds to hemoglobin through an electrostatic attraction. Okay, and the effect of that is that it reduces the affinity of hemoglobin to oxygen. So is this good or is this bad? We'll see. Okay, here's the structure of bisphospho, meaning there are two phospho groups connected to our glycerate. Now, okay, so notice the preponderance of the negative charges. Okay, one, two, three, four, five negative charges in one molecule alone. So you can now imagine to what this would be attracted to. Okay, so it would be attracted to the N-terminal, okay, and the histidine units, which would carry positive charges. And it sits well into the space in between the beta subunits of hemoglobin. Okay, so if it's there, what happens? It forces the beta subunits to be farther apart. What did I say earlier? This is the T state, the taut state, which favors deoxygenated hemoglobin. So again, so histidine 143 and the N-terminal ammonium groups. All right, so what were the experiments? So if you now take hemoglobin and the normal behavior is what was shown earlier. No? So we have this sigmoidal response to oxygenation as a function of oxygen partial pressure. But if you take blood and remove the BPG and check its response to oxygen saturation as a function of oxygen partial pressure, look at what happens. What does this remind you of? This should remind you of the behavior of myoglobin, right? So if there's no BPG in hemoglobin, hemoglobin would function just like myoglobin. But if the oxygen picks up oxygen from the lungs, travels through the circulatory system, and then goes to the muscle cells, and then it won't release the oxygen until there's the partial pressure of oxygen is now very low, then is it still functioning like an oxygen transport molecule? Hardly. Uh, So here's one that's this is another of the secrets, molecular secrets of hemoglobin that's able to that allows it to transport to function as a oxygen transport molecule. Galing, no? So we're alive because of all these molecular interactions. So don't forget your chemistry. Okay, so let's look at the behavior of fetal hemoglobin, a variation of hemoglobin. So in fetal hemoglobin, referring to the hemoglobin found in fetuses, okay, the composition is alpha 2 gamma 2. So the beta subunits have been replaced. What is now the difference? In the gamma subunits, histidine 143 is now replaced by serine. So look at the structure of serine. How is it different from histidine? Okay, so reminder, histidine can be protonated and have a positive charge. serine cannot be protonated, so it cannot carry a positive charge. So what is the effect? The 2,3-bisphosphoglycerate will bind less strongly to BPG. And the effect is again for fetal hemoglobin, shown here by this notation HbF. Again, hemoglobin F fetal. Okay, if it is just HbA, it would be adult hemoglobin. So HbF binds more strongly to oxygen. So here's the oxygen saturation curve of both the fetal hemoglobin, shown in red curve, and the maternal hemoglobin, shown in the blue curve. Okay, so this is our reference. This is what we have: maternal hemoglobin or adult hemoglobin. If there's less bisphosphoglycerate, what did we see earlier? The behavior of the hemoglobin is like that. It becomes closer to that of myoglobin, uh, meaning it binds more strongly to oxygen. So, what happens? Okay, so imagine a pregnant woman having a baby. That's a little bit redundant, but that's okay. So, as the maternal blood, okay, circulates and then goes into the uterus, okay, there's no mixing of maternal blood and fetal blood. No, it's just in the capillaries in the uterus, okay. But the difference in the affinity for oxygen is enough such that essentially there's now a flow of oxygen from maternal hemoglobin into the fetal hemoglobin. No? So maternal oxyhemoglobin transfers its oxygen to the originally fetal deoxyhemoglobin, such that now the such that the fetal hemoglobin is now oxygenated. So once again, I bring back the primary structure of myoglobin from different animals just for us to appreciate. Okay, so let's see. Can I find the histidine F8? So, right, see, so here, in all species, the F8 position will always be a histidine. In all species that has myoglobin or hemoglobin, the E7 will always be histidine. So this will be conserved. So what do you think will happen if for some organism there's a mutation and instead of histidine this becomes like serine? Well, that that organism will probably not survive, no? Because it cannot carry the oxygen. So other effects of changes in primary structure. Okay. We will use sickle cell anemia as an example. So you've heard of sickle cell anemia. What happens is that the glutamate 6 in the beta subunit, this should be a glutamic acid. Okay, it has been replaced by valine. Okay, so what is now the classification of glutamic acid? Okay, it's an acidic amino acid. Valine is a hydrophobic residue. So it will now affect how the subunits interact with each other. Okay, especially under low oxygen conditions, the effect of this is that the beta subunits now link together as stiff rods. Okay, so you can probably imagine again the valine trying to evade, if I can use that term, the polar environment of the cell, and the results will be beta subunit that link together as stiff rods. And then it's enough such that even the whole red blood cell now assumes a sickle shape. Amazing, right? You just have one molecule in the cell, well, one type of molecule in the cell, changing the shape of the whole cell. So it assumes the sickle shape, hence it's called the sickle cell anemia, because under the microscope, the red blood cells now appear like sickle shape. There's a picture. Okay, so the normal disc shape has now assumed that sickle shape. And the trouble with that is that, again, if you normally have that this disc shape, okay, it just allows for smooth flow into our uh, veins, arteries, and capillaries. But in the sickle shape, they get clogged once it reaches the small confines of the capillaries. So there lies the trouble for the affected individual.